stages, that is trophozoites that are always motile and the non-motile cyst. Remember that cysts are non-motile stage of any protozoa. I hope everyone can hear me. So the trophozoite basically is found within the intestinal and the extraintestinal lesions and in dial stools, whereas the cysts are found in non-dial or formed stools. Cyst has got 4 nuclei, which is one of the pathognomic sign of Entamoeba histolytica. So let's see the life cycle. Initially, what happens that contaminated cyst is ingested by the human beings, and when the cyst enters into the intestinal tracts, it is excistation occurs, and it give rise to trophozoites. These trophozoites are motile. They move through the small intestinal system and the large intestinal system, and these trophozoites, they come outside the body and changes into the cystic forms. So you can see here there are different stages. This is the first stages in which the cyst has got the 4 nuclei, which is very visible, and it is the pathognomic of the Entamoeba histolytica. Then cyst is converted, that cyst is ingested, and it is converted into trophozoite. But before conversion, there are 4 stages, that is like excistation. And after excistation, it gives rise to trophozoite. Then trophozoites multiplies, and it gives rise to again different cysts and the trophozoites. And these cysts exit the host body. Trophozoite in cystic form we have already discussed. Initially, what happens that it causes non-bloody diarrhea, but eventually, when the infections becomes more worst, it causes diarrhea with mucus and the blood, along with the abdominal pain. One thing is very important, that there is no animal reservoir, like the host is only the humans. It is more common in homosexual males. And what happens when the trophozoites, they penetrate the colonic epithelium, and they secrete enzymes that produces local ulcers, and these inflammations occur at the site of the invasion. Then when these lesions, they further penetrates and reaches to the muscularis layer of the intestine, large intestine, they produces a typical flask-shaped ulcers. Remember, yesterday we have discussed that in typhoid there is a longitudinal ulcers, but in case of Entamoeba histolytica, there are specific flask-shaped ulcers, and they destroy large area of intestinal epithelium. So this basically Entamoeba histolytica causes invasive disease. Remember that all the protozoa and the bacteria, they are not associated with the invasive disease, but few of them are related to the invasiveness. So I told you that initially clinically there are non-bloody diarrhea, but in severe cases what happens, there is a dysentery, that is a bloody diarrhea, then discomfort, weakness, and one thing is very important, that it produces granulomatous like lesions, that is called the ameboma, right? In those who have infected with this Entamoeba histolytica, but not in all patients, but in some patients it causes granulomatous lesion, that is called the ameboma, and that ameboma usually found at cecal or rectosigmoid areas of the colon. So these lesions could resemble with the adenocarcinoma of the colon and must be distinguished from them. Sometimes what happens that these amebomas look like a colonic cancer, right? So there must be some differentiation. Proper differentiation should be done before diagnosis. Amoebic abscess of the liver is very important. It is usually associated with the weight loss, fever, pain, enlarged liver, and abscess, right lobe abscess that can uh, reach to the diaphragm. You know what is diaphragm? Diaphragm is a muscle that separates chest and abdomen. So it penetrates the diaphragm, and it also causes lung disease. So they have most cases of the amoebic liver abscess occurs in patients who have intestinal amoebiasis. So aspiration of the liver abscess produces brownish pus with the appearance of the anchovy paste. So you can see that this Entamoeba stylotica is very fatal disease. Like if the parasite is present inside any human being, so it can cause very fatal diseases. Huh? Okay. Lab diagnosis may, first of all, the nucleus or nature of the nucleus of the trophozoite is very important, because the Entamoeba stylotica nucleus has got some chromatic granules and centronucleolus, and the cyst has got 4 nucleoli. That is the, I have told you that is the pathognomic. Other tests includes antigen detection, that is a serology, then molecular testing, that is a PCR. In serology, the indirect hemagglutination test is usually performed. Treatments included the metronidazole, flagyl, or tinazole, but hepatic abscess need to be drained. Whenever a patient came with abscess, so the abscess should be drained, then antibiotic treatment will be started.
Excuse me, madam.
जी
मैडम स्लाइड मूव नहीं हो रही आपकी.
हाँ, मैं स्लाइड मूव नहीं कर रही हूँ.
अच्छा ठीक है बस, ठीक है
मैं खुद नहीं कर रही हूँ, दूसरा टॉपिक
हाँ,
शुरू हो जाएगा.
असल में
मैं
स्टूडेंट
इसी
का
के
मैसेज
बारे
आया था,
में
मैंने कहा बता दूं आपको. ठीक है,
नहीं, बच्चों से कहेंगे सुने.
ठीक है
ठीक है. अच्छा जी. प्रिवेंशन में वी आर अवॉइडिंग फीकल कंटामिनेशन ऑफ फूड एंड वाटर, and we will go for good personal hygiene, like good practices of hand washing. That is very, very important. Ingestion of purified water, and but in some cases what happens, there is an outbreak of amoebiasis. So whenever this occurs, the person should be, or the locality should be more confined to the personal hygiene. So in this way we can prevent this disease. Next is Giardia lamblia. Again, the Giardia lamblia, usually all the diseases that is linked with gastrointestinal tract, obviously it is associated with fecally contaminated food. Whenever we eat unhygienic food, whenever we drink unhygienic water, impure water, or contaminated food or water, so it will cause disease. So I think there is not important to tell every time that fecally contaminated food and water will be the causative agents of this diarrhea and the dysentery. Giardia lamblia, basically it is more common in homosexuals, hikers, children who are in daycare centers, patients in mental hospitals. Basically it is associated with you can say that a closed community, people who are in closed who are living or who are having interaction in closed community, they are associated with these parasitic diseases. Life cycle is this, like cyst is ingested, that is contaminated, then exudations occurs, and exudations it is converted into the trophozoites, then these trophozoites are passed into the stool. Again, it has got the two stages, that is infective stage and the diagnostic stage. Diagnostic stage is obviously when the trophozoite and the cyst are found in stool samples, so we can decide that which pathogen is present, or when we go for laboratory diagnosis, like when we take the sample of the stool and we will do serology, or we will do culture. Usually parasitics, they do not grow in culture media, right? So serology or the molecular testing is more preferable and more important. So basically the life cycle of Giardia and Amoeba is almost the same, right? Cysts are found in the formed stools, and for the detection of cysts, 3 stool samples should be taken in both the cases, like in Amoeba histolyticus, because sometimes it happens that we missed the cyst in one sample, so we can find it out in other sample. Again, the cyst has got the 4 nuclei, and the trophozoite has got 2 nuclei, that is here, and it is oval in shape. Giardia antigen or serology is more diagnostic, and string test is usually done. String test, what happens that a string is passed uh, through the mouth, which consists of a weighted piece of a string, and until it reaches the duodenum, the trophozoite adhere or sticks to this string and can be visualized after withdrawal. But this test is not very reliable, and it is usually not practicing. So the very important thing is that Giardia causes malabsorption of proteins and fats. It is not invasive. Remember that Amoeba histolytica is invasive, whereas Giardia is not invasive. In nature, it causes malabsorption of protein and fats. That is very, very important. And this is found worldwide. So it associated with watery, non-bloody, foul-smelling diarrhea, which is associated with nausea, vomiting, abdominal pain, but there is no fever. It means that there is no systemic involvement in case of Giardia. This is the structure of the trophozoite. Here it has got 2 flagella, anterior flagella, 2 posterior flagella. Then it has got a claw hammer, median bodies. This is the caudal flagella. These are the nucleuses, and this is the basal body. The structure is important because sometimes when we looked diarrheal stools, we can find these trophozoite. But for the detection of trophozoite, it is very important that the stool should be fresh, because trophozoite can be find out through their motility. You can see that this trophozoite has got anterior, posterior, ventral, caudal flagella. So all these flagella are helping in movement. So the trophozoite can be find out by the movement of by the movement. As I have told you that it causes foul-smelling diarrhea, watery diarrhea. One thing is also very important, that in case of deficiency of IgA, that is responsible for the predisposition to the symptomatic infection. Malabsorption of fat and protein may occur. That is very, very important. Laboratory diagnosis, I have told you that ELISA detects Giardia, tests cyst, wall, antigen stool, string test, and trophozoites and cyst in both. Treatment involves metronidazole or quinacrine hydrochloride. Prevention, again, prevention is universal. That is infection control practices. Boiled water should be drink. Hand hygiene should be practiced, and there is no prophylactic drug or vaccine available. Cryptosporidium Hominis. Well, Cryptosporidium hominis causes cryptosporidiosis, and it is the symptom with the main system is diarrhea, and it is usually, or it is always, you can say, associated with HIV patients, severe and immunocompromised, especially HIV patient. The former name was Cryptosporidium parvum. The life cycle basically consists of different stages: oocyst, oocyst, and were taken. Contaminated oocysts are taken up during drinking or food, and this oocyst releases sporozoites which form trophozoites. So there are several stages, but we are not going into the details. These trophozoites, they further form schizones and merozoites, and finally microgametes and the macrogametes are formed that combine to form the zygotes. Don't go for, I mean, the life cycle is not very important. It is just showing you, it is just I'm just explaining to give you the idea that how we are taking contaminated food, and how that contaminated food contains different cyst and trophozoites of different protozoa, and when we take this food, these cyst gets converted into trophozoite in our GIT tract. So this is just giving you the idea that how the cyst and trophocyte develop and which one is more infective. So it doesn't mean that you have to remember. Okay, you just understand that what is going on, and it is very well written in the Levinson as well. So although I will share my lectures with you guys, but just understand that what is going on. The most important thing are the symptoms, that which symptoms, like entamoeba histolytica is very invasive in nature. It causes liver abscess, it causes ameboma. Giardia is one of the most important cause of diarrhea, but it doesn't cause invasive diseases. It is associated with non-bloody diarrhea, but it is linked with fat and protein malabsorption. So you should have a clear concept about that which parasite is causing which symptom, and which laboratory diagnosis is important, like the nuclei, four nuclei of cyst of entamoeba histolytica is diagnostic in nature. So you should know that which trophozoite, which cyst is helpful in diagnostic, right? Again, this cryptosporidiasis, the thing is that jejunum is most heavily invested. It is associated with swimming in fecally contaminated. It is like a person who is having regular routine of swimming, so he or she should have a make sure that the swimming pools are clean and it is not contaminated. Basically, cysts are highly resistant to chlorination. Almost all the cysts are resistant to chlorination, but they can be killed by pasteurization or by autoclaving, and can be removed by filtration. Filtration can remove cysts, but the filtration process cannot kill the fish. So the boiling, pasteurization, and the autoclaving could be helpful in removing or in killing the cyst. Autoclaving and pasteurization has got a specific temperature, like in autoclaving 121 degrees centigrade, and I don't remember the temperature of the pasteurization process. So on high temperature, the cyst can be easily killed, but they are not killed by chlorination. In swimming pools, what happens? They clean. Usually, the swimming pools are cleaned by chlorination process. So the swimming pools are one of the cause of spreading this cryptosporidiasis. Laboratory diagnosis may assist in fecal smear by using modified quinone acid fast staining. This acid fast staining is also used for the staining of Mycobacterium tuberculosis. So for the Cryptosporidium hominis, this acid fast staining is also helpful. Treatment will treatment will be done by nitazoxamide drug of choice for non-HIV patient. But in case of HIV or immunocompromised patient, paromomycin is very useful. I hope now everyone can hear me. Okay. Vaccines are not available. Again, prevention can be done by purification and personal hygiene. हेलो जी अच्छा चलिए मैं माइक और करीब करती हूँ ठीक है I think everyone can hear me now. Next is Balantidium coli. Again, it is very identical to Entamoeba stolotica. Cyst is infective, and due to the thick cell wall, it is protected from the acidic conditions of the stomach. The trophozoites are large size, and they have different cytosome, and this is the macronucleus. You can see that, and it has got the cilia. Always remember that trophozoites are motile. They have either cilia or the flagella for the movement, but the cysts are non-motile structure. Live diagnosis may ciliated trophozoite and cyst can be found, but no serological test, like no you can say that no ELISA and no antigen antibody detection can be done in this case. Strongyloides stercoralis. Strongyloides. Basically, the larva penetrates the skin. It usually present in those people who walk barefoot. So the larva, they penetrates through the skin and enters into the lungs. That is very you can say that very dangerous sort of things, because the larva penetrates through the lung and then they move around the lung and then they enter into the GIT tract. So it causes itching and skin infections at the site of entry of the skin, and when it really reaches to the lungs, it causes pneumonia, and then when again it moves around the lungs and enters into the GIT tract, it causes watery diarrhea. Laboratory diagnosis में larva and stool is important, and eosinophilia occurs. Eosinophilia, why eosinophilia occurs? Because in all the parasitic conditions, blood picture always shows eosinophilia. Treatment will include ivermectin and thibendazole, and the prevention, obviously, it is due to the barefoot infection. So proper wearing of shoes and proper wearing of personal hygiene is very important. This is the larva, how it looks like. You can see that there are sharp ends. Necator americanus and Ancylostoma duodenale. These larvae are again, these parasites basically, they causes dual infections. They enters through the penetration of a skin of feet or leg, and they enters directly into the lungs. After that, they reaches to the GIT tract, and these larvae are flariform larvae, right? These flariform larvae are found in the moist soil that penetrates the skin, and they enters into the lungs and then migrates into the alveoli and taken up by the bronchi and trachea and then swallowed. So they develop into adults in the small intestine and attaching to the wall with either nectar or teeth. Basically, nectar means teeth and ancylostoma nectar means cutting plates or ancylostoma means teeth. So in this way they attaches to the wall of the intestine. They feed or they depend on the blood from the intestinal wall, and they lay thousands of eggs that is passes in the feces. So eggs develops into non-infectious form and then into the infectious form. The major clinical sign is anemia, that is a loss of blood, right? And it usually causes microcytic anemia, that is iron deficiency anemia. And then they causes weakness, obviously due to the anemia. They causes weakness, pneumonia, diarrhea, and the pruritic papule they form. These are also called the hookworms, and they are found worldwide, especially in tropical areas. Like so, the walking barefoot on soil leads to the infection, and it is very important public health measures to make sure that all the children, especially, to wear shoes. Proper disposal of waste and the wearing of shoes is prevention. One thing is also important, that prevention is very important, because treatment is not like treatment requires mebendazole and the parental pomade. I'm not focusing more on treatment, but the prevention is very important. The laboratory diagnosis are important, and the clinical picture is very important. Like this in Ancylostoma duodenale nectar americanus, what time the filariform larva penetrates the skin. So you should have a clear concept about what is the filariform larva, and this is the stage of larva which enters into the skin. So you should have a clear concepts about that. Okay, this is the male and female picture of the Ancylostoma duodenale. This is the egg of the Ancylostoma duodenale. Then Ascaris lumbricoides. Ascaris lumbricoides, again, the humans are infected by ingesting warm eggs in food or water contaminated with human feces. The egg hatches into the small intestine, and the larva migrate through the gut wall into the bloodstream and then into the lungs. They reaches the alveoli, then the bronchi, trachea, and then again swallow. Within the small intestine, they converted into the adult, right? And they live in the lumen, and they do not attach to the wall, right? So the adults are the largest intestinal nematodes. Remember that they are the largest one, often growing to 25 centimeters or more. So it is also called the giant roundworm. Thousands of eggs are laid daily, that is passed in the feces. Major damage occurs during larval migration. As I have mentioned, that these larvae they pass through the respiratory tract as well as the GIT tract. So during the migration, they causes major damage, right? So the primary basically site of infections are the lungs, where there is an eosinophilic exudate occurs in response to the larval migration. Most infections are asymptomatic. Ascaris pneumonia with fever, cough is with heavy larval burden is common. Abdominal pain and even obstruction can result from the presence of adult in the intestine. Here are the laboratory diagnosis: presence of oval eggs. The eggs has a typical appearance. It has got oval shape with irregular surfaces. Mebendazole and parental pomade, the drug of choices. It causes pneumonia, malnutrition, intestinal obstruction, and prevention is again proper disposal of feces, contaminated food should be avoided, proper hygiene is very important. Now viruses. Up till now we have studied different important protozoas. Now viruses. Rotavirus and norovirus are the two important viruses that are associated with diarrhea, and they are linked with childhood diarrhea. Like the diarrhea, they are more common in kids, in children's. The one thing is very important, that usually viruses are associated with acute diarrhea. They are not linked with chronic diarrheal stages. So children's between 6 months and 24 months of age are more infected, and they are more common in hospitals and daycares. Rotavirus basically is a double-stranded RNA viruses, and it belongs to a family Reoviridae, and they are, like I've told you, they are more notorious for causing diarrhea in children's. And what they do, they destroy intestinal cells, that are the enterocytes. So that is very important. One thing is all that every children in the world is infected with a rotavirus at least once by the age of 5. So you can imagine that how severe or how destructive it is. Immunity usually develops with each infection, so when next infection occurs, so that infection could be less severe. Adults are rarely effective, and it has got 9 species. So that is A, B, C, D, F, G, H, I, and J. But the A is the most common species that causes more than 90% of rotavirus infections in humans. Rotavirus E, which is seen in pigs, has not been confirmed as a distant disease so far. Again, the virus is transmitted by orophycal route. It damages the cells of the small intestine and leads to gastroenteritis, that is called stomach flu. It has no relation with the influenza. It is associated with hospitalization. Those children who are hospitalized are more prone to develop this diarrhea. And it also, the one thing which was very amazing for me when I was preparing this lecture, that this rotavirus is pathogen of livestock as well. So it is not causing infection in humans, but it's causing infection in livestock as well. So most of the rotavirus infections occurs in developing countries. So as we know that developing countries are usually have very poor hygienic conditions. So the people of developing countries are more prone to develop different infectious diseases. Rotaviral enteritis could be mild to severe disease that is characterized by nausea, vomiting, watery diarrhea, and low-grade fever. Its incubation period is about 2 days before symptoms appear. Symptoms usually acute, as I have told you, that viral cause of diarrhea is in acute condition. Dehydration is very common, and rotavirus infections usually can occur throughout the life. The first usually produces symptoms, but subsequent will be asymptomatic. And due to the immunity acquired in children, so adults are not susceptible to rotavirus infection. Detection are usually done by serology, because we are not culturing viruses, and so the serology enzyme immunoassays they are standard for their detection. And there are different types of electron microscopy and PCR, reverse transcriptase polymerase chain, RT-PCR, can detect and identify these viruses. RT-PCR is because it's the RNA virus. Next pathogen is norovirus. Norovirus is responsible for half of the outbreaks of diarrhea, and again it is the causative agent in the children, and it is associated with acute diarrhea. It is a very contagious and that causes vomiting and diarrhea, and it can infect people of all age group. It is not like associated with the particular age group, but people of all age group can associated with this diarrhea. It causes non-bloody diarrhea, that is a watery diarrhea. Abdominal pain is very common in case, and usually people have, those who are infected with the noroviruses usually have contact with already infected person. So the touching contaminated surface, then putting unwashed hands in mouth, so all these are the way of transmitting. The thing is how to prevent these viral infections. So first of all, again, wash your hands often, rinse your food, fruits and vegetables before eating, cook shellfish thoroughly, stay home when sick and for 2 days after symptoms stops. Avoid preparing food for others when sick and for 2 days after symptoms stops. The outbreak is very common. The virus spreads very easily and quickly from infected person to others through contaminated food and surfaces. So outbreak can happen anytime. So it is very important that the person should take care of themselves and others as well. The viral disease are usually self-limited. They complete their incubation period and they subsides. They do not need to take any any medicine, especially antibiotics or antiprotozoal medicines are not for viruses. Viruses are self-limiting diseases. That is very, very important. But here we are practicing giving antibiotics to the person who is having viral infection. So these were the references from where I have prepared this lecture. I will share my lecture as I told you earlier. Now we move towards a question and answer session. So any question, please? Okay. What is the difference between invasive and non-invasive? Invasiveness is basically when the microorganism, either it could be bacteria or it could be protozoa or it could be viruses. What happens whenever they enters into the GIT tract, they attaches to the epithelial lining. They attaches to the epithelial lining of the GIT tract, and some pathogens they just attach, but they do not penetrate. But in case of Entamoeba histolytica, I have told you that they penetrate the muscularis layer of the intestine and form their toxins, which further causes ulceration and necrosis and formation of abscess. So invasiveness means that whenever the pathogens penetrate through the epithelial lining and causes destruction, and other like Giardia doesn't causes any invasiveness. It is just attaches to the epithelial lining, releases their symptoms, and then that's it. It means that it is not entering into the epithelial lining. Fungi are usually not related to the gastroenteritis. Very rare, and if they are associated with gastroenteritis, it means the patient is highly immunocompromised. So far we have studied that bacteria and protozoa are causing infection causing infection in immunocompetent patient as well, but fungi in diarrhea is not associated with fungi. If it is associated, that it means the patient is highly immunocompromised. Yes, larva by walking barefoot at seaside, yes, it is found in larvas are found on moist soil, so you can anybody could have this infection while walking barefoot. Acha ji, next question. Adenoviruses, yes, adenoviruses are also causes diarrhea. Excistation means that the cyst converted into the trophozoite. So this process is called the excistation, right? Like a cyst has got the 4 nuclei. Each nuclei give rise to, or the each cyst they exist, that is converted in trophozoite, and each cyst give rise to 4 trophozoites, right? Deficiency of IgA will predispose the function to, yes, obviously, because IgA is usually present in the secretory. It is basically the secretory antibodies that is present inside the body. So if the and it also prevent attachments of the pathogens to the epithelial cell linings, right? So if the IgA is absent, obviously it causes it also predisposes to infection. Normal flora could cause infection. Swimming pools are not safe. Swimming pools are not safe, especially I don't know, there must be a proper system of cleaning of the swimming pool other than chlorination. They should change the water after some times, whole water after some times. So that could be one of the preventive effect of prevention from Cryptosporidium, because we cannot pasteurize or boil the swimming pool, na? So I guess that there should be a system of replacing the water with fresh water, because in most of the swimming pools what they do, they just fill the swimming pool with chlorine, and excess amount of chlorine is also very hazardous for the skin and for the hairs. So swimming pools should be properly cleaned by changing the water as well. No, non-invasive, or it could be non-invasive organisms are always toxigenic. No, sometimes what happens that the actually invasiveness, non-invasiveness, and the number of bacteria. In my yesterday's lecture, I told you that the number of bacteria is very important. If the number of bacteria are less and they are non-invasive in nature, so it will be flush out from the GIT tract through 1 or 2 loose motions or loose stools, right? But in case of greater number of bacteria along with the invasiveness, so it will cause dysentery, bloody diarrhea, foul smelling diarrhea, even watery diarrhea. So number of bacteria are very important. No, Giardia is not the only cause of foul smelling stools. There are a lot of other causes as well, but we have discussed Giardia. Homosexuals are more prone to dysentery because the homosexuals are people are devoid of personal hygiene. So right from the beginning, I am repeating this thing that the personal hygiene, hand washing, infection control practices are very important. So whenever there is a breach in personal hygiene, so it will lead to any infection other than diarrhea. So that's why homosexuality is one of the most important homosexuals are the most important people, because they are devoid of any personal hygienes, right? Actually, viral diarrhea is the period of viral diarrhea is self-limiting. It usually most of the viral diarrheas are subsided by 7 to 10 days. Usually 7 to 10 days. Watery diarrhea will have trophozoite. Cyst will be formed in non-watery diarrhea, in formed stools, because watery diarrhea you always have a trophozoites. No, they are so small, they are so tiny that sometimes you can't see them, na? So they will make a small hole in your skin and they just enter it on through it. They don't need to make a big cut. Amoebic dysentery required metronidazole, that is flagell. Psychogenic diarrhea are associated with inflammatory bowel disease, or usually what happens that there is a proper mechanism of our sympathetic nervous system in our GIT as well. If you remember your physiology of gastrointestinal tract, so what happens whenever we are in panicky condition, when we are in tense, so this sympathetic stimulation causes our GIT system to be more hyperactive. So what happens? There are two conditions occurs, either diarrhea or constipation. It is not necessary that always diarrhea can occur. Constipation can also occur. So it is the one of the the nervous system. Our nervous system is stimulated, our gastrointestinal nervous system, that further leads to the overactivation of the GIT tract, that leads to the psychogenic diarrhea. And it is very, very common, especially in students. I'm sure you people have gone through this psychogenic diarrhea during your examination period. I'm sure. Okay, G. Any question? Yeah, Viva or even in exams, I had psychogenic bouts of psychogenic diarrhea during my profs. I can always remember. Okay, it's a wonderful session today, and I'm very happy that you people are very interactive and you are asking questions. So I'm very, very happy with you guys. Please be interactive in my next 2 lectures that will be coming up most probably next week. So that's a wonderful class I have ever seen. And please contact any of your CL through email. I'll give you all my lectures. I'm always available. You can ask me any question, any problem associated with any examination point of view or anything. I'm always there. So don't shy to contact me, and always be interactive like this. Thanks. Okay. I will be waiting. I will share all my lectures, like acute and chronic inflammation, and these diarrhea and dysentery part 1 and part 2. So if you will email me, I'll send you all my lectures, right? Any question? Any question? Should we stop this class? No, don't go into depth. Don't go into depth. Try to study my lecture, right? And then you can consult with Levinson, that is 15th edition. So that is more than enough. Don't go for life cycles. Life cycles are just for the explanation. Don't go for life cycles. I have told you that prevention is important, because it is also important for generally. You should know that how the personal hygiene is very important, because diarrhea is one of the most common disease. It is one of the most common infectious disease. So you should know the prevention. Prevention is very important. I don't think that you should remember the name of the drugs that are useful for the treatment. You should know the sign and symptoms. You should know the specific pathognomic identification. Like in case of entamoeba, I have repeated, there are 4 nuclei in cyst. Again, in Giardia is non-invasive. It causes fat and protein malabsorption. So these are the specific things that are associated with each organism. So just do that. Don't get into the depth of each and everything. And if you find any problem, I am here. You can ask me. I think we have to stop this session.